Dear students, today we shall be talking about the characteristic features of the series Pisces. Actually, we are going to talk about the fishes in general. Before moving on, it is my request to you all to like and subscribe my channel BioLearnia. Now, how we are going to define a fish? The simplest of all the definitions, while defining a fish, is that the fishes are aquatic, cold-blooded vertebrates respiring with the help of gills and locomoting with the help of the fins. This is the simplest ever definition of a fish. But most of the fishes are streamlined, though there are many exceptions which I shall be talking later on. Now, when we talk about the diversity of the fishes are diverse of all the vertebrates. They are almost more than that of the other vertebrates together. That is, the total number of the mammals, birds and reptiles and amphibians, they are lesser than that of the fishes. And one reason the fish are so diverse is that 70% of the planet is covered in water. The animals in this group live in a variety of habitats, ranging from the coral reefs and kelp forest to the rivers, streams, and open ocean. Another is that the fish are very old on the evolutionary scale. According to the fossils records, they have been on this very earth for more than 500 million years. So, that was the Devonian era is called as the golden age of the fishes. The total number of the living fish species, that is about 32,000, is greater than all the total number of the other vertebrates. Now when we talk about the habitat, we say that the fishes are aquatic animals. They are either freshwater or marine. Now, it is not only that they are occupying the fresh water and the marine water, they are present in the brackish water as well. But if they have the capability to live in the fresh water and the marine water, it means they can be present in the brackish water as well. The brackish water has a salinity lesser than that of the marine water and more than that of the fresh water. They are present in all the extreme habitats. They can be present in the hot thermal geysers and they can be present in the cold, chilly, Antarctic and Arctic regions as well. They are found in the higher altitudes. They are found in the deeper part of the oceans as well. They are found in the deepest of the caves, and they can be found in the marshes and wetlands as well. So there are different varieties of the fishes occupying different habitats. In a simple way to say that wherever there is water, there is a fish. Now, talking about their covering. When we talk about their covering, the skin is usually covered with scales, either scales. Now, these are the scales. They can be covered with the denticles, or they can be covered with the bony plates as well. Now, integument in some of the fishes, they are nagged. It means they don't have any kind of the scale, denticle, or bony plates present. So, their integument contains mucous glands. The integument also contains the chromatophores in some of the fishes, so that they have the ability for the camouflage, so that they can escape from the predators and they can capture the prey as well. Body shape. Mostly the shape of the body of the fishes is streamlined, but some are elongated and some can be dorsoventrally flattened, as in the case of the flatfishes. When I say streamlined body, it is a kind of a shape that lowers the friction drag between a fluid like air and water and an object moving through that fluid. Now, when I say streamlined body, it is mostly narrower toward the anterior side and the narrower toward the posterior side. It is because of this narrowness, it is according to because of this pointed regions, the fish can easily swim with less and less friction. Now, when I was talking about the locomotory organ, the locomotory organ in the fishes is fin. Now, fins are the locomotory organs of the fish. They can be paired and they can be unpaired. And they are supported by the soft spiny rays. Soft or spiny rays which are dermal in origin. Now, for example, there are certain fishes which are paired and there are certain fishes which are unpaired, such as this very dorsal fin. It is an unpaired fin. We call this very kind of fin as the median fin. The caudal fin, it is the unpaired fin. Now we do have some other fins as well, such as anal fin, which is also a median fin. It is also unpaired, whereas the other fins such as pectoral fin, such as pelvic fin, they are paired fins, and they have a combination of, these very fishes have the combination of medium and thus they have the combination of median and paired fins. They have medium dorsal, anal, caudal fins. There is a pair of pectoral fin and a pair of pelvic fins located posteriorly. The expanded part of the fin is supported by the thin skeletal rods known as the fin rays. Now these are the fin rays. These very skeletal rods. These are the fin rays which support the fins. Now some of these very fins are very soft, whereas some are hard. So some of the fins we call them as the soft rays, and some of the fins we call them as the spiny rays or hard rays. So, talking about the tail, this is the caudal fin and it is the muscular and used for the propulsion. Though there are many kinds of the caudal fins or the tail fins. It can be rounded, it can be forked, it can be indented, it can be truncate, it can be lunate, it can be pointed. But the function of this very caudal fin is propulsion. It propels the body forward. Nostrils. These are the nostrils. These are the paired nostrils. So the nostrils are present one on each side. They are paired but do not open into the pharynx except lungs. So it means that these very nostrils, they are the external nostrils. They don't open into the pharynx inside because their role in the respiration is limited, because the role of the respiration is taken over by the gills here. So in these very fishes, the nostrils, they are not internal, they are external, except the lungfishes. The lungfishes have the internal nares as well. Now, when we talk about the barbels, now these very barbels, these we call them as the excellent tactile organs. In the form of the barbels are hair-like structures. They are present in some, and mostly they are present in the bottom-dwelling fishes. Now these are used as a tactile organs so that they can feel their prey. They can feel the movement of the water. They can feel their prey. So mostly the catfishes have the excellent barbels present on the anterior region, that is on the head region. That nasal barbels can be present, maxillary barbels, nasal barbels can be present, maxillary barbels can be present, then mental barbel can be present. So there are different kinds of barbels present in the fish. Now when we talk about the endoskeleton, endoskeleton is either cartilaginous or bony. Now here there are two main kinds of the fishes. Now cartilaginous fishes and bony fishes. The cartilaginous fishes are mostly belonging to the chondrichthyes, whereas the bony skeleton is present in the bony fishes. So when we talk about the endoskeleton, it is mostly cartilaginous in the case of the chondrichthyes, whereas it is bony in the case of osteichthyes. Modern sharks, when we talk about these very modern sharks, they most likely they have evolved their lighter cartilaginous skeleton to become faster swimmers, because they are not as massive as the bones. They are lighter than that of the bones, so that they can evade predators and swiftly catch their prey. So this helps them for the easy movement, faster swimming, and to move easily. So the loss of bone in their skeleton is also supported by the fact that the oldest and the most basal of all the jawed vertebrates, that is the placoderm, had heavy skeleton. So the most recent phylogenetic analysis of the lower vertebrates, the placoderms, they appear as being basal at the common evolutionary level to the sharks. So it means that these very sharks must have evolved from the bony fishes, so that they can be the stronger predators, they can be the masters of the sea. And you all know that the sharks, we call them as a swimming noses, and they are the topmost predators of the marine environment. So this is their cartilaginous skeleton, which makes them so successful, because it helps them to move easily. They are robust, they are lighter, and their endoskeleton is not so massive. When we talk about the respiration, the organ of the respiration is gills. These are the gills. This is the arrangement of the gills. These are the gills of the teleost, or the bony fishes. So these are the gills, these are the respiratory organs, though there are certain accessory respiratory organs also present in some of the fishes, that can be in the form of the air sacs, that can be in the form of the suprabranchial cavity, or it can be in the form of the, you can say, aboruscent organs in the case of Clarius, and it can be tubular air sac in the case of Heteropneustos fossilis. Now, when we talk about the gill cover, this very cover which is present on the gills in the case of bony fishes, we call it as the operculum. So we also call it as the gill cover. This is the gill cover in the case of the bony fishes, whereas in the case of chondrichthyes, or we can say in the case of sharks and the flat fishes, these very slits, they are not covered by the operculum, that is, they don't have any gill cover, but they are open in the form of slits. Now, when we talk about the slits, they are 4 in the case of bony fishes and 5 in the case of the chondrichthyes. Mostly 5 in the case of chondrichthyes. Though they can vary from the 5, 6, and 7. 6 in the case of Hexancus and 7 in the case of Heptancus. Here, I was talking about the gill slits, that they are mostly 5 pairs. In the case of cartilaginous fishes, and 4 pairs in the case of bony fishes. So this is very important point, that the 4 pairs of the gill slits are present. Now here, these are the slits. Now here, the slits are internal, that is inside the gill cover. So this is a diagram which is not having a gill cover. The gill cover is taken up to show the gill slits. So here in the 4 gill slits are present, it means there are 4 gills which are having the slits in between. But here in the case of sharks, the 5 gill slits are present, but they can be 6, they can be 7. 6 in the case of Hexancus fish and 7 in the case of Heptancus fish. Now another organ, that is the swim bladder. The air bladder, it is also present, but it is secondarily lost in some fishes. As we all know that this is the swim bladder. The main function of this very swim bladder is to maintain the buoyancy. It is also connected to the internal air, so that it can maintain the buoyancy as well. It is filled with the nitrogen and other gases as well. Now, when we talk about the heart of the fish, it is two-chambered. It is two-chambered heart. So one auricle and one ventricle. So this is the atrium, and this is the ventricle. So only one auricle and one ventricle, though there are accessory chambers as well. We call them as the sinus venosus and conus arteriosus, that is the conus arteriosus and sinus venosus. In some cases, the bulbus arteriosus can also be present. So when we talk about the cranial nerves, there are 10 pairs of the cranial nerves. That is ophthalmic, olfactory, platinus, maxillaris, mandibularis, hyomandibular, glossopharyngeal, branchialis, lateralis, that is visceralis, and buccalis. These are the 10 pairs of the cranial nerves which are present in the case of fishes, whereas in the case of the mammals or human beings mostly the 12 pairs are present. So when we talk about the internal ear, I was talking earlier when I was talking about the swim breeder that they have only one internal ear which is present in the case of fishes. So our ear and the brain, that is in the case of the humans, they translate vibrations into sound and language. But fishes hear, but their ears are on the inside, that is on the inner part. So bony fishes detect vibrations through their ear stones. Now what are the ear stones? The otoliths we call them as the ear stone. Both people and the fish use parts of the ear to help them in balancing. They act as a balancing organ. So here what we are seeing, that nothing is outside, nothing is seen outside, nothing ear like structure is seen outside, but yes, this very internal ear, this is present on the inner side. So it is made up of the sagitta, it is made up of the lagina, lapillus. So there are different structures, there are different you can say bones which are associated with it. So talking about the kidneys in the case of fish, it is mesonephric. When we say pronephros, it is the earliest nephric stage in the case of human beings, and it constitutes the mature kidney in most of the primitive vertebrates. Metanephros arises the caudal to the mesonephros at the 5 weeks of the development. It is permanent and functional kidney in the higher vertebrates. It is derived from the intermediate mesoderm. When we talk about the mesonephros, it is in the Greek, meso means middle. So it is one of the three excretory organs that develop in the vertebrates. It serves as the main excretory organ of the aquatic vertebrates and as a temporary kidney in the case of reptiles, birds and mammals. So when we are talking about the basic fish kidney, so this is the figure showing the mesonephros kidney and having a mesonephric duct which opens into the cloaca. Now looking here at the schematic diagram, wherein this is the earlier stage of the kidney, that is the pronephros, and then the pronephros degenerates, and then it is the mesonephros which takes over the function of the kidney, or which acts as a kidney, and then in later stage it is the metanephric kidney which takes over in the case of higher vertebrates. But in the case of these very fishes, it is the mesonephros which is present as a kidney. Talking about the gonads in the case of fishes, the gonads possess the true gonaduct. The sexes in the case of fishes are separate and the development is indirect. Though there are certain exceptions such as grouper fishes wherein they are hermaphrodites, and in the case of fishes development is direct. It means that the larval stage is there, that is that very stage which does not resemble the adult stage is there. That is why we call this very development as the indirect development. And thirdly, the amnion and the allantois are absent in the embryonic stages. When we say amnion and allantois, these are the mostly the cowrings which are mostly produced in the higher vertebrates, that is the reptiles, birds and mammals. So these are absent in the case of lower vertebrate, that is the fish and amphibians. So in the case of fish and amphibians, they are mostly called as the anemniotes, whereas the higher vertebrates we call them as the amniotes, because in the higher vertebrates amnion is present, whereas in the lower vertebrates amnion is not present. Now the reason is that that these very organisms, that is the lower vertebrate, that is fishes and the amphibians, they lay their eggs in water, so there is no worry about the desiccation. So that is why the amnion and the allantois, they are that is absent in the case of the organisms which lay their eggs in the water. Whereas in the case of terrestrial organisms, there is the problem of the desiccation. So that is why their eggs have a covering of the amnion and the allantois. So this was all about the fishes, the important characteristic features of the fishes. Hope you enjoyed the lecture, and it is my request to you all again to like and subscribe my channel, that is Biolearnia. Thank you very much.